Okay, so this is my recording. What we just talked about seems to work better this way for me. We're going to be talking about CRISPR and what we've been doing in lab, and I'm going to try to bring all these pieces together for you so you can see what we were actually trying to do. Okay, so I've added this resource to the Moodle. It's about, I don't know, it's a document, it's a PDF. It's from AdGene. It's called Desktop Guide to CRISPR, and it has everything written out. This would be essentially the text for this type of material. It's very well written with many figures. I took some of my figures from it. You can find other ones in there if you want to see other things in more detail. It's very detailed and very up to date. It's called AdGene's Desktop Guide to CRISPR. So from the beginning, I've talked about the fact that CRISPR is a lot about the design. That's a huge, tremendous piece of it. More than, I mean, most molecular biology experiments involve a lot of design because you're dealing with, they're a little bit abstract. You're dealing with sequences. You visualize them as letters. Ultimately, they're becoming protein products and stuff. CRISPR is very much the same way, but it involves lots of more pieces that need to finally come together to work together. And so this shows here, this emphasizes this point. So this planning and design phase is a big piece of it. Then you put your CRISPR reagents into your system, be it mouse or E. coli, and do the CRISPRing, and that's this little guide that's done at the end. Okay? So just remember, I think the themes to hang on to as we go through here, that the CRISPR starts with a targeted double-stranded cut in DNA. And that targeted double-stranded cut is made by the combined power of using the Cas9 endonuclease and the fact that it's targeted to cut with a guide RNA. And so we can pretty much pick a guide RNA of our choosing, express it with a Cas protein, and get a double-stranded cut. Once we have a double-stranded cut, then there's possible outcomes. And that's the genome editing part. So again, here is a schematic of what the Cas9 with the guide RNA looks like once it's assembled in vivo. So remember, in order for the complete cleavage to occur, you need this 20 nucleotide target region here. It needs to be adjacent to a PAM site. And then it's followed by some more RNA. That's called the RNA scaffold. And so we need, when we want to target our CRISPR, we want to determine our target that's next to a PAM site and then make sure that whole thing is expressed with this guide RNA. And that stabilizes the Cas9 on our DNA target the way we want it to. So this is the part that we have control of, this target. And so if we can find a target that we want to cut, it's adjacent to a PAM site, we can cause Cas9 to cut there. And once we have a double-stranded cut there, then what's going to happen in the cell? Cells don't like their DNA to be damaged. So the cell is going to repair it. It's going to either repair it with homology-directed repair or non-homologous end joining. In this material here, they tend to refer to homology-directed repair. I called it homologous recombination. Same thing, okay? Homologous directed repair, HDR. Equals homologous recombination. With the holiday junctions and the strand invasion, we went through DNA repair purposefully so we'd understand these steps when we see them in relationship to CRISPR. Okay, so what's next? So once we've had the cut, actually, now we'll kind of talk about the types of editing that can occur after that. Okay, so again, it all starts with the cut and the targeted cut with the Cas9, which is indicated here. So the first thing is we have a single targeted cut someplace in the genome, and we don't put in a repair template, just do the cut. The natural way that'll be repaired in mammalian cells is non-homologous end joining. E. coli has a thing called alternative end joining, which is not so efficient. When non-homologous end joining occurs, it's not very accurate. It can cause the introduction of small indels, which means addition of up to 10 nucleotides in either subtraction or addition of up to 10 nucleotides. And because they're not a multiple of 3, you've disrupted the gene, you're probably going to have some kind of frame shift mutation, right? A small indel would cause a frame shift mutation. And somehow disruption of your gene. So that's untemplated repair. It's just You targeted the cut, and then the DNA repair machinery just does something to get the DNA glued back together. Another step up from there is you can target 2 different cuts, okay? So these could be 50 base pairs apart. They could be a kilobase apart. The farther apart they are, the frequency of repair will be a little bit smaller, and we'll talk about how to glue them back together. But basically, if you put in 2 cuts, and let's maybe we make some different colors here. So we'd have this bright blue region here, and then we had this green region here. If you put in 2 cuts, when they glue back together, you'd have your green region get glued together with your bright blue region, and you'd have taken out the intervening sequence. And so that could be a whole exon, that could be a whole much larger piece of a gene, so you could have a larger deletion there. So now let's look at the C through G. So now we're going to talk about taking control of the repair process. So the first 2, we've just used Cas9 and a guide RNA to cut in 1 or more spots. And that's pretty handy and has lots of experimental applications. Now we want to take it to the next level and actually control the repair. And we're going to control the repair by adding a repair template. Okay, so repair template. So what do we do with that? And the type of repair template dictates the type of repair. So we have a lot of flexibility in that. So a very simple type of repair would be putting in a small insertion. So remember, when we see an oligo, we're talking about something that may be 20 to 40 nucleotides long. Usually not much longer. So you could use an oligo that's adjacent to your cut site, and as it starts to activate non-homologous end joining, this oligo will get added in there, and you'd get a small insertion. There's plenty of reasons you'd want to do that. This depends on what type of repair you're trying to create. You could also use a single-stranded oligo with a point mutation in it. So in this case, look, we've cut here. We actually want to repair that spot and change the nucleotide sequence. And we may want to create a specific point mutation to cause this very specific change to a gene. For instance, we could design this oligo so that it's changing a coding sequence to go from a serine to an alanine, for instance, right? It's a single point mutation or a cluster of point mutations. Okay, serine to alanine, encoded by the DNA in this oligo. So we put in a point mutation because we used a repair template. And when it gets repaired, now we're starting to use homologous recombination, or one version of it here. And so we've added a repair template which directs a single point mutation. We can also use oligos to make deletions. Again, by the virtue of the fact that the oligo is short, the deletion would be smaller, but it would still function as a deletion. So again, if we use this blue and green code, hopefully we'll see what happens here. So there's going to be homology in the oligo. One side of the oligo is going to be to the blue. One side's going to be to the... Oops, I'm saying this is actually a deletion. I'm looking at I did it backwards here. Sorry. Let me stop. Well, I'll just fix it. Let's erase that and make that right. Okay. Correction. So in this case, you can see this with the red. We'll still use our blue and green homology. We can do a small insertion by an oligo. So we'd have homology here on our oligo to the blue. This part would be the blue part. We'd have homology to the oligo. And this is on either side of the cut site to our green part. And then we have new material that are stuck on the oligo, made in that oligo on a machine. It doesn't involve much labor. You just order it. And you add, so after the cut, when this repairs, you've introduced material into the genome. The flip side of that is if your cuts are farther away. So this is really, you'd have two cuts here. You could have a repair oligo that span the gaps of those two cuts and then brings together, when they repair, it causes a deletion. So let's say, well, like it's sort of like this guy up here, but using an oligo to blend, to facilitate. So it'd be kind of taking B, but using an oligo to help drive the repair that invokes this deletion. Because if I use a homologous recombination, it's a little more efficient and not as random. It's templated now. And also you may want them to fuse together in a very specific way. If you're using non-homologous end joining, you'll get this deletion, but there will be some variability at the junction. If you fuse the ends together using homologous recombination, you'll get a much more precise junction. It'll be specific, especially let's say you want to delete a particular exon in a gene and fuse two other exons together but keep them in frame. You can't risk that deletion causing a frame shift. So you'd want to make sure this repair is very accurate. And related to that is insertion of larger constructs at the span larger than the length of DNA that you'd be able to put on an oligo, and this is what we did. Okay, so we had our targeted site that cut, and let's say now this represents LAC-Z. This represents LAC-Z right here. We can cut in LAC-Z, and then and then the LAC-Z is here and here, and then in the middle here is where we're gonna, in our repair template, we've introduced GFP. So homologous recombination drives the repair at this end. There'll be a holiday junction here, and drives repair at this end, and once on either side of the cut, and in so doing it brings GFP into the middle. And that's, you need to do that on a plasmid because that is too large. Our GFP was 900 base pairs long, and we had arms only about 70 or 80 base pairs long, but sometimes you might want to design with arms a little longer. So once you get above 100 or so, it's easier to manipulate this. 100 base pairs, you'll need a plasmid. So we can control the cut with the guide RNA, and we can control the repair with the repair template, and that's the editing part. That's our genome editing. Those are the steps. Now we're going to go through them in some more detail and hopefully see how we were attempting to do that in lab. So again, this just is another launching off slide. So basically, we're going to talk about now is editing, knockouts, and editing. And again, power of CRISPR is that it starts with a targeted double-stranded cut, and that double-stranded cut is relatively easily generated by building a guide RNA that can interact with Cas9. And so if we just cut and repair without much repair template, let in-house repair enzymes do their thing, we'll cut, and then there'll be a repair, and we'll have some type of disruption in the gene we want to study. Or else we take it to the next step and we add a repair template and make it. When we add the repair template, then we're saying, okay, not only are you cutting here, we want the edited product to look how I planned it. So I skipped this while I talked. I'm going to skip it now. Just another look at the setup and the alignment. Okay, I must go up to here. So I'm going to get to our what we did in lab. Okay, so again, it's about the planning and the designs. Now I'm going to go through those steps a little more slowly to hopefully see what we did related to this big process. And again, remember, this planning and design phase is a big and large important piece of doing CRISPR. It's the biggest part of it. So let's talk first about if we just want to make a single cut someplace in a gene. What are the steps involved to doing that? Okay, so basic steps. We just want a single cut, non-homologous. The repair will be driven by non-homologous end joining, which we know is sloppy. It's not as accurate. It's untemplated. It's not accurate. And it'll result in some type of indel, which would cause a disruption in our gene. But there's some randomness to this. So what do we need to do? What are the steps involved? We need to know what gene we want to study. We want to know what organism we're working in. So we need to know our genome sequence. We want to know the gene. And then within that gene, we need to figure out where we can target. So there's certain criteria for a guide RNA. So then we figure out what that looks like, and then we need to have some way to clone it. And so sometimes we're going to talk about that in a second. And then we need to get the guide RNA and the Cas9 active protein active in the cells. And then when they're active together, we're going to have a genome. We're going to have a little guide RNA here. We're going to have Cas9 making the cut. Okay, so let's talk about that with specifics, with a specific reference to what we were actually doing. So, what was our organism? Let's do one. Our organism is E. coli. Okay, what gene did we decide to target? LAC-Z. And actually a specific piece of LAC-Z that we know we're sure overlapped with the E. coli genome. Okay, so within LAC-Z, we had to select a guide RNA. So what are the criteria for picking a guide RNA? It needs to be 20 nucleotides long. And it needs to be adjacent to a PAM site, which for the Cas9 that we're using, the PAM site is an N, G, G. Okay, so now we picked the target, but now we need to do something so that we can have this DNA, PAM site DNA, be expressed as a guide RNA in the bacteria. So in the case of E. coli, the best way to do that is actually to make the guide RNA as a plasmid. And there's a promoter on it. So when it's in E. coli, the promoter drives the production of an RNA transcript. Okay, so that means that's why we had to use that p-target plasmid, and we'll look at that again in a second. Okay, transcript. So then once we made this plasmid with our PAM site and our guide RNA target in it, we need to get them into E. coli. So what we did in the lab, because it's E. coli, we did a bacterial transformation. Right, and that's the same thing you do to get any kind of DNA molecular material into bacteria as a transformation. Okay, so in this case, if we're just doing the cut, let's abbreviate that there. We put our pCas plasmid in. Into bacteria here. Whoopsies. And our target plasmid. Once those are in E. coli, again, we'll induce transcription of Cas9 to make the Cas9 endonuclease. And this p-target plasmid has a constitutive promoter, so it's going to be making RNA. So we'll have our guide RNA in E. coli, and we'll have the active endonuclease. Oops. Okay? And so when the Cas9 is in the presence of the guide RNA, it's going to cut. So let's draw, here's a million base pairs of E. coli DNA. Here's LAC-Z in the middle of that million base pairs. And then we're going to express our guide RNA. It's going to bring Cas9 to the middle of the LAC-Z gene. and it's going to cut. And then, because non-homologous end joining or alternative end joining in E. coli is sloppy, there'll be some mutations introduced, some indels, some small additions or insertions. Joining, sloppy, creates small mutations called indels. So we should have some type of disruption to our functional laxity. Okay, so that is just doing one-step targeted cutting and spontaneous repair of CRISPR, using CRISPR. So the next thing we did is we wanted to take it up a level, and we tried to do these simultaneously. Maybe in a lab with more time, we could have done them one at a time. So the next step is not only just to have not just to cut with Cas9, and then target the cut with your guide RNA. We now want to control the repair. So that means we need to make a repair template. So now with a repair template, it allows us to control the repair. Okay, so now we're going to do this again with the repair template in the context of our thinking specifically about what we were trying to do with E. coli. Okay, so 1, what's your genome and your sequence? E. coli. LAC-Z. Our guide RNA needs to be 20 nucleotides near a PAM site. And that's the same as before. Okay? But now we got to think about our repair template. So remember, we talked about where the PAM site is where the Cas9 actually cuts. It doesn't cut at the NGG. It cuts 3 nucleotides into the target sequence. So our cut is going to occur here. And if we say that this is LAC-Z, we'll make it blue. We want to design a repair template around this cut site that's going to allow us to introduce GFP. So we need to think specifically about where we're cutting and then how we want to insert GFP there. And we can do that using Gibson cloning. There's no restriction sites there. So now we're at a little bit more complex level of design here. So now we're thinking about designing this repair template, but it's got to be compatible with where we're cutting. Okay, so the repair template has to be compatible with where we're cutting. Compatible with cut site. Okay. So we spent some time in lab trying to discuss and design the Gibson cloning to do this. So what we did in order to do Gibson cloning, we're going to start with our we're going to use the PUC19 as the source of our arms for the repair. So remember, I think I have the plasmids coming up actually. Let's see if we can get them. There they are. Right. Okay. So remember, so these were the plasmids we had to work with. So let's concentrate. Now we're just talking about PUC19 and PGLO, the PGLO plasmid with our various GFP mutants in it. Okay. So right in here, Inside of PUC19 is the LAC-Z gene, and that is where we're cutting in E. coli, but it also is represented on this plasmid. And there's a spot right in the middle here where we picked for our guide RNA. And after we've cut open at that guide RNA, we want to do a repair where we use LAC-Z and we fuse GFP into the middle of it. Oops, let me switch to green there for our GFP. There. So that's going to be our repair template. Okay? And the way we did this with Gibson cloning, we can make all these pieces by PCR. Okay? So what we need to do is PCR our GFP out of pGlow. So we're going to have primers that are at either end of the GFP to amplify it. And then in order to get the backbone that we need this as the coding sequence of GFP needs to be moved into PUC19. We want the PUC19, all this other stuff to be there. So we want the ORI to be present. We want ampicillin to be present. So we actually amplify in the opposite direction to make our backbone. Okay, so we're turning PUC19 into a linear molecule by amplifying at our cut site. We know where it's going to cut when we do CRISPR, and that's where we want to introduce the GFP. So we're going to use PCR to amplify at the cut site, but going in the opposite direction. So now we're think of opening that PUC19 up into a linear molecule at the same place our guide RNA will eventually cut in the E. coli genome. And so then once we have those two pieces, we have our PUC19 kind of backwards and opened up here, so it still has AMP on it, right? Still has AMP on it. It still has part of LAC-Z on it here where we opened it up. It still has an ORI, right? Okay, so we have that PCR product, and then we're going to have our GFP PCR product. And I'm just going to draw it long for the sake of making this a little easier to neater on this slide here, but it's obviously a little shorter. And that's our GFP PCR product. And we design them in such a way that we know we create an overlap at the edges. So we made sure that when we did this PCR, we actually added a little GFP sequence to the edges of the PUC19, and we added some LAC-Z sequences to the edges of the GFP. So when we put them into a Gibson reaction, they circularize, and then we wind up with GFP inserted right where we want it, in the middle of LAC-Z. But this is not in the genome yet. This is just the template. This is now, we've made a repair template. It's a GFP repair template. That's a LAC-Z GFP repair template. Okay, so let's see. So then we need to get all of that into E. coli and allow the repair to occur, right? Let's see what comes up next. So this is just a reminder of what, because I showed you this before, and you can reference this here if when you're thinking about it again. So P-target-F allows the expression of our guide RNA in conjunction with the scaffold RNA, right? Remember there's that extra little section on it called scaffold. So remember, what we needed to do is put our little LAC-Z strip in there, and then the rest of the target plasmid allows for the expression of that little extra bit of RNA that has to interact with Cas9. So this is our little LAC-Z specific fragment in right adjacent to where it needs to be to target. There's the complement of the PAM site is in this scaffold, and that helps line up the guide RNA for cutting. We used PUC19 because it has LAC-Z in it. So that was our reference source sequence for cloning, and also it gave us the backbone to make our repair construct. We used pGlow, our mutant pGlows that we made in the first half of lab. So we had a source for our GFP to introduce into to make our repair template that we just talked about. Then the last plasmid we need to use is this pCas, and we only use that at the end. That's the one that expresses Cas9. And we only express it when we're ready to induce the cleavage by the guide RNA and the subsequent repair. I put this in here. I know it probably looks a little more confusing when you start seeing the kind of names and the nucleotides, but I list them, and I want you to pay attention and try to make sure you understand not what the sequences are per se, but I have actually tried to illustrate what the purpose of each one is, okay? So, for instance, we have the GFP LAC-Z. So when we actually get to do this, now we're no longer little arrows on the paper. We do need to think about nucleotides in the long run. And in order to make our repair template, we used Gibson cloning. Okay, so we wanted to make this and you can see that here we put GFP in the middle of LAC-Z. So these oligos, one amplifies the GFP with LAC-Z tails, the other one amplifies the PUC19 with GFP tails. And if you go back to that other slide, you'll see how that's how we need to blend those together to make our repair template. So this is for Gibson cloning of our repair template here. Okay, but of course the repair template doesn't do any good unless we've cut. So where do we make our guide RNA template here? So that is shown. The oligos we use for that is this very long oligo here. And so these 20 nucleotides in the middle represent our target site in LAC-Z. And remember, we need to get, I showed it a couple times like this. I can't go backwards in this version of filming, but that LAC-Z target site needs to be put into the target plasmid adjacent to the RNA scaffold. And so you can backtrack on the other slides and see that. But we didn't have to PCR that. We could just order the oligo and then recombine that into the target plasmid. So that was making our, so these oligos were for making our guide RNA. Okay. So those are the main reagents that we talked about using. So this is the questions that are in your lab archives that we answered together in this last lecture. I want you to go through and answer them again on your own, and we can go and we'll review our results together on Friday, what everybody's answers are. Hopefully this, as you answer through things, I've tried to do it very stepwise so you can see each piece and how we used it along the path. Because granted, it is a little tricky. So this is the first steps of the questions are more about the design component. So there's about 20 questions there. The second half of the questions are what we expect to see once we put all the different reagents into E. coli. So this is putting everything into E. coli and inducing Cas9. So this is putting everything in. This is our little E. coli cell here. And so remember, one of the things we're doing is we're trying to disrupt the endogenous LAC-Z gene. The endogenous LAC-Z gene is just as good doing blue-white as the LAC-Z on a plasmid. So the only functional LAC-Z that it will be in any of these things will be in the E. coli genome or not if we disrupted it. And then have we successfully been able to introduce a fluorescent protein into the LAC-Z gene. And when we walk through these things, you should see, hopefully we did this together and you can do it on your own again. Cas9 is required. So imagine the result. If there's no Cas9, are we going to get CRISPR? If there's incorrect targeting, are we going to get the right kind of CRISPR? Right? So this helps you think about what reagents you need in a reaction and the expected outcome. And we'll go over that together on Friday. And then also remember in that separate file, I want you to try to redesign an analogous type of CRISPR editing setup, just using slightly different genes. So you have to kind of think about it from afresh. And we'll go over that on Friday as well. And then hopefully we'll understand CRISPR at least a lot better than we did on Tuesday. Okay, I'm going to say goodbye.